example, you are a psychologist and, I don't know, some institution, and a drug company sends you these two very persuasive advertisements. Unfortunately, they're not terribly persuasive in the way that they're viewed right now on the screen because they're a little bit fuzzy. But the first one is an old man with a cane, and it's an advertisement for a drug called Thorazine, and it's to control the advertisement says senile agitation. All right. And the second one is a drug called Nembutal, and apparently it's given to children to calm them down, perhaps for a very long time. So you have some suspicion that these drugs may have some effect on levels of anxiety. Okay, so that's one of the effects that you would like to check, whether or not these drugs and what kind of an effect these drugs have on levels of anxiety. But you also have another suspicion, and that is that the time of medication, the time that you take the drug, whether or not you take a particular drug with food or without food, may also either enhance or, in contrast, have just a different effect on the level of anxiety. So if you take a drug with food, for example, you might suspect that it will be metabolized much quicker, right? And it will have simply more effect of the drug on the behavior. So ideally, what you would like to do in this experiment is to study the effects of both the type of medication, Thorazine and Nembutal, and the time of medication, whether or not you're taking it with food or without food. Okay? So this is that exact table that I showed to you before. And let me just advance here manually. So you have these 6 conditions now, and in 6 conditions, you have the type of medication, which is the first level would be placebo, taking placebo. The second level is Thorazine. The third level is Nembutal, and you can be taking the placebo either with food or without food. The drug Thorazine with food or without food, and the drug Nembutal with food or without food. And in each one of the gray cells, you have an average of this behavioral measurement that you've chosen to measure the level of anxiety. It could be some kind of a psychological test, or I don't know, something that measures behavior. Is this example clear? So basically, the idea is that you, in a multi-factorial experiment or in a factorial experiment, you have to have all of the combinations of these different levels. Okay? So the number of conditions is going to be determined by the number of these combinations. That's all that I really wanted to show you here, and now we're going to try the full screen mode again and see how it works.
Attention, this is the engineer. Our fire alarm test is concluded. Thank you.
Well, we're very lucky today. Okay, so having spoken about factorial design, I'm just going to very briefly review what kind of factorial designs there are. So you must be familiar already that there is a between subject factorial design, right? And that means that each gray cell in this table here is a different group of participants, right? And you have a within subject design as well, where any participant in your experiment actually takes part in all of the conditions in your experiment, right? So you have one group of participants and they participate in every single condition you have in your experiment. Okay, any questions about this? This should be all pretty familiar to you. Okay, so let's do a little comparison, a side-by-side comparison of the between subject and within subject experiments. Why would you want to use one over the other, for example? So let's say you want to have an average of 5 data points in each one of these gray cells that I showed you before. So each one of these gray cells, you want to have an average of 5 data points. In order to do that, in order to achieve that in a between subject design, that would mean that you would need to have a total of 30 people, right? So it's 5 data points, or 5 people, or 6 in each condition, and there are 6 conditions in total, 30 people total. In a within subject design, you can still have those same 5 people, right? So it seems like the within subject design is a lot more economical. There is actually another reason why you may want to have more people in the between subject design. So remember, the purpose of any experiment is to really compare and contrast, right? Compare and contrast your conditions. If you want to talk about the influence of a particular independent variable, you want to see the difference between the conditions, right? Right? Yeah? Okay. So if we are talking about the between subject design, the difference between these conditions would be, first of all, due to some treatment effects, right? And by treatment effects, I mean the effects of the independent variable. But it could also be due to the difference between the groups. So, in fact, this difference between conditions consists of these two parts. And sometimes this difference between groups is going to go against the effects of your independent variable. So in order to be able to see the difference between different conditions that is due to treatment effects alone, you may want to, in that particular case, you may want to have much larger samples in each one of the cells, right? So that these differences, these incidental differences due to just differences between groups are just going to average out, right? In the within subject design, this is not a problem. You always have the same group in every single cell. And what this means is that ideally, the difference between cells, or the difference between conditions, is really going to be due to treatment effects alone, to the effects of your independent variable. Any questions about this? No? Okay. However, if you remember this also from your exam, there is a reason why a within subject design, it also has basically certain limitations, and those limitations are order effects and effects of sequence. If you remember, order effects have to do with absolute time, right? So when, in the duration, total duration of your experiment, the person is doing a particular condition, when are you taking the measurement? So if you have an experiment of 6 conditions, the measurements that are taken in condition 6, that is at the end of the experiment, may be affected by variables such as fatigue, or perhaps learning. So, for example, in your experiment, if you're doing math problems, some sort of math problems in every single condition, there needs some learning, some transfer of skill from one condition to the other. Okay? And then there's, of course, the effects of sequence. And these are probably more traditionally identified as a sort of transfer problems, or transfer of skill problems. So you can imagine that if you're doing a math problem, a difficult problem followed by an easy problem, your performance might be different if you started doing an easy problem first and then went on to doing a more difficult problem. You may have learned something from doing an easy problem, right? You may have gotten a gist of some difficult mathematical concept that you were able to apply them in a difficult problem. So that is a transfer of skill. And there are ways, of course, to take care of these debilitating is the word correct? I'm not quite sure. But basically, debilitating. You may be able to take care of these debilitating effects by using counterbalancing. And what I'd like you to do is, for the last part of today's lecture, when I'm going to be presenting an example experiment, I would like to think about how we address these different limitations of an experimental design in our experiment. Okay?